Good morning. Today we are going to see competency number 5.2, properties of cardiac muscle. And I'm Dr. Devi Nithiya, Associate Professor of Physiology, Sri Lakshmi Narayana Institute of Medical Sciences. So first, the learning objectives. In this session, at the end of the session, the students should be able to list the properties, physiological characteristics of cardiac muscle, their electrical and mechanical properties, and also the ionic basis of the action potential of cardiac muscle. So first, going into the properties of cardiac muscle, I would like to brief to you that there are two types of cardiac muscle fibers based on their anatomical positioning. That is, the muscles which are present in the atrium and that in the ventricle. So they have some amount of difference in their characteristics. And physiologically speaking, they are once again classified into two types. That is the excitatory and the conductive muscle fibers. The excitatory muscle fibers, these are present in the pacemaker tissue, like the SA node and AV node. And the conductive muscle fibers, these are present in the His bundle Purkinje fibers. So the physiological characteristics of cardiac muscle can be studied under two settings. So the first setting is a beating heart. So it means that the heart beats and the properties of the heart, beating heart can be the following. That is the automaticity, rhythmicity, conductivity, contractility, excitability, and distensibility. The second setting is of quiescent heart. Quiescent heart means the heart is quiet, so it is not beating. So it was not beating continuously, or the impulse generation is not spontaneous. So if you give a stimulus, it will respond to it. Okay, so such a setting is called as the quiescent heart. Here the heart is actually dissected out and kept inside a nutrient broth so that the tissues don't die. As well as when you give any stimulus, they'll react to it. So the properties which can be studied in a quiescent heart are the long refractory period, functional syncytium, extrasystole and compensatory pause, all or none law, Stark's phenomenon, length tension relationship, frequency force relationship, and load velocity relationship. So we'll be seeing each one of this one by one. First, what do you mean by a dinner weighted heart? So in a dinner weighted heart, you cut off all the nerve supply to it, but the perfusion of the cardiac muscle is maintained. So what do you expect when the nerve supply to the heart is cut off? What do you expect? You generally expect that the heart is not getting stimulated or there is no impulse generation, but this is not so. There is spontaneous impulse generation within the cardiac muscle. That's because of the presence of the pacemaker tissues. So this property, where there is spontaneous generation of impulses in the cardiac muscle, even when the nerve supply is being cut off, is called as automaticity. Okay, so this is one of the most unique properties of cardiac muscle. So the hierarchy of these pacemaker tissues are as follows. So high up in the list is the SA node, followed by the AV node, then the His-Purkinje fibers, and the ventricular muscles. So what brings about this hierarchy? It's basically the number of impulses which are produced spontaneously. Okay, so here you can see that the SA node will be producing the maximum highest number of impulses. The details of this we'll be seeing later. Then the next is property is rhythmicity. So what do we mean by rhythmicity? There is a regular discharge of these impulses in a rhythmic fashion. So the primary pacemaker of the cardiac tissue is the SA node, and this produces impulses at a rate of 60 to 100 beats per minute. When talking about the hierarchy of the pacemakers, we already saw that the SA node is the one which is at the highest hierarchy, and this has an impulse generation of around 60 to 100 beats per minute. So now let us try to understand this with the help of a animal experiment, that is a frog heart experiment. So what is being done is, please concentrate on this diagram. So you can see that this is the ventricle and the lower portion is the right atrium and the left atrium here. So the frog heart differs from that of a human heart in having only 3 chambers. So it has 2 atria and 1 ventricle. Okay, unlike that of humans where we have 2 atria and 2 ventricles. So here in this diagram, there is a ligature which is placed here. Okay, the stanius ligature 1. That is, in between the sinus venosus and the atria, a ligature is placed, and that is called as the first stanius ligature. The sinus venosus in frog's heart is similar to that of the SA node in the human heart. So here we are trying to block the conduction from the SA node to the atria. Okay, that is the sinus venosus to the atria. So let's see how is it reflected in the frog's heart activity. Okay, so normally coming onto this graph, you can see that the frog heart beats in this fashion. So when the first stanius ligature is applied, you can see that the heart rate, the distance between each of the beat has increased. So that means that the heart rate has decreased. Okay. And now along with this, if you try to apply another ligature, that is the second stanius ligature, which is in between the atria and the ventricles. And now referring to this graph, you can see that the impulse generation is further delayed. You can see that the peak of each of the contraction, there is a distance between those contractions. Yes. So in case of second stanius ligature, this distance is further increased. So this denotes that the heartbeat further decreases. You can also notice that the strength of the contraction also decreases with the first stanius ligature and the second stanius ligature progressively. So the primary pacemaker of heart in human heart is the SA node, which produces impulses at a rate of 60 to 100 beats per minute. Then the secondary pacemakers are the AV node, his bundle, Purkinje fibers, and the ventricular muscles in the given order. So you can see that gradually the number of impulses which are produced by each of these tissues is lessened. So first is the SA node, then comes the AV node, the bundle of his and Purkinje fibers, and later is the ventricular muscle. So when the ventricular muscle acts as a pacemaker, you get a very low heart rate of around 15 to 30 beats per minute. And normally with the SA node as a pacemaker, we have a normal heart rate of around 60 to 100 beats per minute. So who determines the heart rate? And at which rate does this heart beat? Okay, so this is just an analogy. If you see, there is a person driving the car. Okay, he's trying to pull a motorcycle, and that once again is connected by a chain to a bicycle, and that to another person who has a skater under his foot. So whatever be the speed of this person who is using a skater or a bicycle or a scooter, if the car drives at a particular speed, all of them will be pulled at the same speed. So who determines the rate here? It is the car. The speed at which the car moves determines the speed at which the entire setup moves. If in case this chain between the car and the scooter is cut off. Okay, just like how in first stanius ligature where the SA node is cut off. Now what happens is the scooter which was riding at 40 kilometers per hour is going to determine the speed of the setup. Okay, so this is just an analogy to explain how the hierarchy of pacemaker tissues determine the heart rate of the heart. Then coming on to the next property of conductivity, it is the impulse which is generated at the SA node has to be propagated to the other parts of the heart. And this impulse propagation is possible because of the property of conduction of the cardiac muscles. So now looking into the conductive system of the heart. So what are the components that you have to know here? So you know that the impulse generation occurs at the SA node, which is the primary pacemaker. So from the SA node, the impulse has to be carried on to the AV node. So how does this happen? So this happens with the help of the tracks. Okay, there are 3 tracks which carry the impulses from the SA node to the AV node. So what are these tracks? They are the tracks of Bachmann, tract of Wenckebach, and the tract of Thorel. So these are the 3 tracks between the AV node and the SA node. So do they all conduct the impulses immediately and without any delay? So this is about the speed with which it is conducted. It is generally noticed that there is some amount of delay at the AV node. What is the reason for this delay at the AV node? It's because of the small size of the nodal tissue, the branching cell pattern, the slow voltage calcium channels, and also less number of gap junctions at the AV node, which cause a delay in the conduction of these impulses which are produced at the SA node. So is it a problem that there is AV nodal delay? No. It is in fact has a very important physiological significance. That is, this delay helps in better filling of the ventricular chamber. Okay, so that the ventricles get filled adequately during this AV nodal delay. Next, talking about the conduction velocity. So the speed with which the impulses are conducted across the different tissues is as follows. Though the, and this is in terms of meters per second. Okay, so here the SA node conducts at a speed of 0.05 meters per second. The internodal pathways at a speed of 1 meter per second. AV node at 0.05 once again. Then the His bundle and ventricular muscles at once again at 1 meter per second. Here you can notice that the Purkinje fibers are the fastest conducting tissue and they conduct the impulse at a speed of 4 meters per second. So a small video to explain the conducting system. So you can see that the SA node impulse generation, these impulses are carried to the AV node and from the AV node to the bundle of His and the bundle branches, the right and the left bundle branches, and then to the Purkinje cells. And finally, it reaches the base of the heart. So once again, SA node, AV node, then to the bundle, and then to the branches, left and the right, and the Purkinje fibers. Next property is the contractility. So the impulse that is generated at the SA node and the one which is propagated through the different tissues which we saw in the previous slide, these are nothing but the events which occur because of the electrical activity. Okay. Now this electrical activity is continued or it is projected as a mechanical event which is observed as the contraction of the various chambers of the heart. So contractility is nothing but the mechanical event which occurs after the electrical event of impulse generation and propagation. The fifth property of cardiac muscle fibers, it is the excitability. So what do we mean by excitability? It is the ability of the cardiac muscles to respond to different stimuli. So what are the stimuli that it can respond to? It can either be an electrical stimuli where the concentration of the various ions act as the stimulus, or it can be hormonal stimulation in the form of hormones which cause increase in heart rate, decrease in heart rate, increase in the contractility, decrease in contractility. Like some of the hormones which are involved are catecholamines, adrenaline, okay. So then thyroxine. So all these hormones are involved. And then also the state of autonomic activity. So autonomic activity, we mean the sympathetic and the parasympathetic activity, which also alters the excitability. When the sympathetic overactivity occurs, there is increased excitability of the cardiac muscle tissue, muscle fibers. And when there is parasympathetic overactivity, the excitability decreases. Then the sixth property is distensibility. What do you mean by distensibility? It is the ability of the cardiac muscle to stretch. So this denotes compliance. So as the volume inside the chambers increase, the pressure does not rise sharply. This is because the chamber can increase in its size. So this is called as the compliance. So this helps in the atrial and the ventricular filling. Okay, and this plays a very important role in determining the end diastolic volume. Importance of end diastolic volume we'll be seeing in our later classes. So that was the property of all the beating heart. So all those properties can be studied in case of a beating heart setup. Now next we'll move on to the properties of quiescent heart. First is a long refractory period. So what do we mean by a refractory period? Refractory period is a period during which the muscle does not evoke a response, no matter how strong the stimulus is. Okay, whatever stimulus you give and however strong the stimulus might be, the muscle tissue will fail to produce a response. So that period of non-reactive reactivity is called as a refractory period. So for the cardiac muscle, there are typically two reactive periods that can be recognized. So one is the absolute and the other is the relative refractory period. So what is this? So here you can see that when time is drawn along the x-axis and the cardiac muscle action potential is drawn along the y-axis. So in this case, if you see this pink line or the red colored line graph, this represents the electrical activity, that is the action potential. So you can see that the membrane becomes depolarized, and then there is a plateau phase and then a repolarization phase. Okay, and the green line graph, this represents the mechanical activity. Okay, so this duration where the depolarization and the plateau phase of the action potential occurs, you can see that the contraction has started and has reached a peak. During this period, in the initial 200 milliseconds, whatever stimulus you give, it is not going to cause another stimulus in the muscle fiber. Cardiac muscle fiber will remain absolutely refractive to the stimulus which is given. Then next part, if you see this blue shaded portion, it is called as the relative refractory period. Here, the muscle fiber goes in for the repolarization phase, and also the mechanical activity, the muscle is in the relaxation phase. Here, if you try to give a strong stimulus with repeated frequency, there are chances that the muscle fiber will react to it. So this is the refractory period which I have to mention. So this was for a cardiac muscle. Whereas for a skeletal muscle, if you see, the action potential, there is a sharp increase in depolarization, and a very steep depolarization which occurs immediately. So the action potential as such ends before the peak of the mechanical activity happens. Okay. So in this case, you can see that the absolute refractory period is very short in skeletal muscles when compared to that of cardiac muscles. So this is itself a very important feature of cardiac muscle fibers, and this helps in preventing the tetanization of the muscle fibers. So what is that? We'll see in further details now. So here, if you see, I have denoted the time scale along the x-axis, and along the y-axis we have the action potential, which is from minus 70 millivolts to plus 20 millivolts. Now, if you follow the period of absolute refractory period, and this is the relative refractory period. So this red line denotes the electrical activity. There is a depolarization followed by a plateau phase. So this part of the electrical activity contributes to the absolute refractory period. So here you can see that the mechanical activity which is achieved is the contraction has begun, reached its peak, and it is starting to relax. So these events which occur in the initial 200 milliseconds is contributing to the absolute refractory period, where whatever stimulus you give, there is not going to be any increase in the response or any further production of response, new response. And now the rest of the part of the repolarization phase, which coincides with the relaxation phase of the mechanical activity. This is called as the relative refractory period. So the long refractory period of the cardiac muscle fiber prevents in the tetanization of the heart. Okay. So even if you give continuous stimulus, the heart muscles will not react to it, and hence tetanization of the heart muscle is prevented. Now moving on to the eighth property of cardiac muscle, that is the functional syncytium. So functional syncytium is nothing but the whole unit working as a or functioning as a single unit. All the structures together working or functioning as a single unit. This is possible because of the gap junctions. Gap junctions are junctions through which the ions can pass very easily. Okay. So the atrium functions as a separate syncytium. The ventricle functions as a separate syncytium. Why do they have to function separately, atrium and the separate unit, the atrium and the ventricle? This is because when the atria contract, the blood from the atrium will enter into the ventricle. So this will aid in the ventricular filling. When the atrium contracts and when the ventricle also contracts together, ventricular filling is not possible. So when the atrium contracts, you want the ventricle to relax so that the blood from the atrium can enter into the ventricle. So this will help in ventricular filling. Moving on to the next property, that is extrasystole and compensatory pause. So here, when you give a sufficiently strong stimulus during the relative refractory period, there occurs a extra contractile response. So here in this graph, if you see, when you give an extra stimulus, an extra systole occurs. Okay. Once this extra systole has occurred, it is followed by a pause. This is called as a compensatory pause. Okay. This is called as a compensatory pause. So zooming it. Here you can see that the first three contractions are the normal contractions where stimulus is given and the response is recorded. Okay. During the extra during the relative refractory period, when you give an extra stimulus, you can see that there is an extra systole or an extra contraction which is observed. Okay. Following this, there is a pause. This pause is called as a compensatory pause. Okay. And this compensatory pause is also followed by a PEP. Okay. That is a increase in the potential. Okay. So the next is the all or none law. All or none law. What do we mean by all or none law? It is the magnitude of response of the tissue to the stimulus will remain the same irrespective of the strength of the stimulus. That is, if you give a subthreshold stimulus, you will not get any response. Okay. If you give a threshold stimulus, you will get a response. And if you give a supra-threshold stimulus, once again, you will get the response which was obtained for the threshold stimulus. That's all. Okay. So there cannot be an increased response or a decreased response. Either the response is absent or the response is optimal and which is present for both the threshold as well as the extra threshold, supra threshold stimulus. So all or none law is the nature of the action potential. Okay. And this will also help in the syncytial property or the nature of the cardiac muscles, so that the atrial contraction occurs as a unit and the ventricular contraction relaxation occurs as a separate unit. Now moving on to the 11th property, that is the staircase phenomenon. So this is studied with the help of the Steckenstein's ligature, that is the ligature which is present in between the atrium and the ventricle. So obviously it is studied in a quiescent heart. So what we try to do here is we are giving repeated stimulus at very short intervals. That is, at intervals less than 10 seconds, you continuously keep giving the stimulus. So what happens is that the magnitude of the contractions will slowly increase for the first 3 to 4 contractions. Okay. So why does this happen? And does it not violate the principles of all or none law? Because we saw in the previous slide that when you give a stimulus, it either is present, it either elicits a response or there is no response based on the strength of the stimulus. But here what are we doing? It is each stimulus will alter the conditions of the next stimulus. Okay. In case of staircase phenomenon, the interval at which you are giving the stimulus is going to be very short with less than 10 seconds duration. So when you do that, each of the stimulus will alter the presetting for the next stimulus. So to further explain what is the mechanism behind the staircase phenomenon, it increases the cytostolic calcium levels inside the cytoplasm. What is this? When the muscle fiber goes for contraction, the calcium concentration will increase, and when it is in the cytoplasm, and when the relaxation phase occurs, this calcium will enter back into the sarcoplasmic reticulum, right? But since in case of staircase phenomenon, you are giving continuously short intervals, at short intervals, you are continuously stimulating the tissue. So you don't give enough time for the calcium to enter back into the sarcoplasmic reticulum. So that is causing an increase in the cytosolic calcium ion concentration. Okay. Because of this, there is increase in the contraction. Also, because of the repeated stimulation, there is increase in temperature and enhanced enzymatic activity. And the increased temperature will also decrease the sarcoplasmic viscosity. So all these factors aid in progressively increasing the contractions for at least 3 to 4 contractions when the stimuli is given at short intervals of 10 seconds. Next, moving on to the length tension relationship. What is this length tension relationship? It is nothing but the Frank-Starling's law of the heart. Here the force Sorry, there was an interruption in the net connectivity. Yes. Now we were talking about this length tension relationship. So what is Frank-Starling's law of the heart? It is the force of contraction is directly proportional to the initial length of the muscle fiber within the physiological limits. So what does this mean? Here, if you consider the length of the muscle fiber along the x-axis and the tension developed in the muscle along the y-axis, you can see that there is a passive tension which is present at the resting state. So as the length of the muscle fiber increases, the tension will gradually increase for a particular level up to the optimal length. And then if further the muscle is stretched, there is a rapid increase in the tension. So this we can, it is similar to using a rubber band. Now initially when you try to stretch the rubber band, up to one particular level, it is going to be a slight increase in the tension. But when you pull it to a further extent beyond an optimal length, the tension which is created in the rubber band will be higher. So similarly here also passive tension up to a particular level it will increase, will increase gradually, and then after a particular level there is a sharp increase. Okay, so this passive tension is recorded during the diastolic phase of the heart. And next is the active tension. So active tension, if you see, it increases with the increase in the length of the muscle fiber, reaches a peak, and then beyond the optimal length, it starts to decrease. So this is studied during the systolic phase of the heart. So why does this happen? So initially, when the muscle fiber is getting stretched because of the filling of the chambers with blood, there will be increase in the length of the muscle fiber, that is increased distensibility, and also the tension in the muscle fiber will keep increasing. Beyond one particular level, the muscle fiber will give way. Okay, so that there will be a decrease in the tension in the muscle fiber. So summating the two, that is the active tension and the passive tension, you will get the total tension which is observed as and gradual increase in the tension with the increase in the muscle fibers. Okay, so this is the total tension which is developed. So sarcomere length, how does it react differently at an optimal length and beyond the optimal length? At the optimal length, the maximum interaction between actin and the myosin filaments occur, and therefore there are maximum cross bridge formations. So when the cardiac muscle stretch occurs, there is an increase in the sarcomere length, and this increases the affinity of troponin C to calcium. Whereas beyond the optimal length, when the muscle fiber is stretched, that leads to decreased interaction of the actin and myosin filaments because they are pulled further apart. Okay, so the interaction between actin and myosin will decrease, and there will be decrease in the cross bridge formations. So this is the reason why you can see that the pressure changes in the active phase will decrease. The tension decreases when the muscle is stretched beyond the optimal length. So coming once again to Frank-Starling's law, what does it state? It states that the force of contraction is directly proportional to the initial length of the muscle fiber within the physiological limits. So this part of this statement, that is within the physiological limits, is very important to be noticed. So what is responsible for this Frank-Starling's law? So there is there are four mechanisms which can be described, and the first of it is increase in the end diastolic volume, that is the total amount of blood which is present inside the ventricles at the end of the relaxation phase. Okay, so that will cause that determines how much stretch of the ventricular muscle has occurred. Okay. So when the stretch has increased, that will increase the actin and the myosin bridge formation, and that will lead to an increase in the force of contraction. So we can see that the length of the muscle fiber, as it increases, the force of contraction also increases. The second mechanism which is described is when there is an increased stretch of the muscle fibers, there is increased opening of the stretch sensitive calcium channels. When the calcium channels are opened, we know that the calcium entry into the cell occurs, and therefore the cytosolic calcium levels increases. Once the calcium concentration increases inside the cell, this contributes to an increase in the force of contraction. So this is mechanism 2, which is aided by the calcium channels and the concentration of calcium inside the cell. And the third mechanism is based on when the increased cytosolic calcium levels, this not only stops with the calcium which is entering from outside to inside the cell, but it also causes an increase in the release of calcium ions from the sarcoplasmic reticulum. So this further adds on to the concentration of calcium inside the cytosol. So furthermore, this increases the force of contraction. And mechanism 4 is when the myocardial stretch occurs, this will enhance the affinity of troponin C to calcium, and there will be an increased force of contraction. So that was the Frank-Starling's law of heart and the mechanisms which help in Frank-Starling's law being established. Okay, next, talking about the frequency force relationship. This is also called as the Bowditch phenomenon or the Trippy phenomenon. So when there is an increase in the heart rate, there is an increase in the force of contraction. This is mainly because of the accumulation of intracellular calcium concentration. So how does it happen? When there is an increase in heart rate, there is an increase in the number of depolarizations per minute. That is, the number of stimuli are more. Okay, impulse generation is more. So when these depolarizations are more in number, they will cause more of calcium entry into the cell. Okay, during the plateau phase of action potential, and because of this, there is a decrease in the duration of the action potential, and there will be an increase in the force of contraction. So even if the duration of the action potential is short, because of the presence of increased levels of calcium ions, the force of contraction increases. So this is called as the Bowditch phenomenon or the Trippy phenomenon. Also, the increase in heart rate will diminish the intervals between the heart. Because of this, the increase in the inner calcium current per depolarization occurs, and that once again will lead to an increase in the force of contraction. And finally, coming on to the load velocity relationship, the relationship of velocity of shortening to the load imparted on the muscle with which the muscle contracts. So this is mainly going to give you a relationship between the velocity of shortening, which is denoted in the y-axis, and the load which is imparted on the muscle fiber. Okay, here if you see, if the velocity of contraction will be inversely proportional to the load. So that's why you see that the line is a downward graph. Okay, so as the shortening is faster with lower loads and slower with higher loads. So this denotes that the shortening will be faster when you have less load to act against. Okay, and slower when there is higher loads which are imparted on the muscle contraction.